Hello everyone. This is Dr. Sanmitra Ghosh from the Department of Biological Sciences, School of Life Science and Biotechnology, Adamas University. I welcome you all to my first online class on immunology, and today we are going to have an idea about the immunoglobulins and the antibodies. So there will be a series of classes on this topic, and today is the introductory session or the first class on antibody. So to begin with, let's have an idea about what the immune system is and what is the role of the antibodies or the immunoglobulins in this particular immune system. Now, the immune system actually consists of a large repertoire of different types of components with very unique properties or unique strategies to combat the effect of the foreign molecules which continuously attack us, attack our body. So there are lots of weapons, lots of arms and immunities with which our immune system is actually well equipped, with the help of which they can fight against the indefinite number of foreign molecules that we encounter in course of our lifetime. And broadly, this immune system can be, or immune response can be divided into two basic response. One is the first line of defense, that is the innate immune response, which we are born with, which is actually not very specific, and it is the same for all the members of a single species. On the other hand, we have the acquired immunity, and in course of the lifetime of an individual, of a human being, as he or she is born, till the end of his or her life, as they are confronting different types of foreign molecules, foreign components, and they are interacting with them, they are able to identify which one is good for them and which one is bad for them, and which does not have any effect on them at all. So when they are able to identify this potent, harmful agents in our surroundings, that is by means of the particular type of immune response, which is known as the acquired immune response or adaptive immune response, because we are adapted to this type of immune response in course of our lifetime, in course of our experiencing with the by confronting with the foreign molecules. Now, diverse type of components are there in this acquired immune response or the adaptive immune response, with large number of organs, large number of cells involved in it. The major cells which are involved here are the lymphocytes, and these lymphocytes can be of two different types, the T lymphocytes and the B lymphocytes. And this two type of lymphocytes and the different components that we derive from them, they actually give rise to a huge amount of unique type of immune response, as I mentioned at the very beginning also. And they can be categorized as the humoral immune response and the cell-mediated immune response. Now, in today's chapter, as I mentioned, that we are going to have an idea about the immunoglobulins or the antibodies. Here, the immunoglobulins, they form a part of the humoral immune response. So to begin with the chapter, to have an introduction towards this antibodies on the immunoglobulins, let me first share my slides with you. Hope the slides are clearly visible to you. We will begin with the definition of the antibodies. Or better to say, if the question is asked, what is an antibody? Here is the answer. Antibodies, which are also known as the immunoglobulins, they are actually some proteins or glycoproteins that can bind the antigens, and they are present on the surface of the B cell membrane in the form of the B cell receptor or BCR, and they are secreted by the plasma cells in response to the antigen stimulation. Once the naive B cells, the small naive B cell, which is in the G0 phase, it interacts with the antigen. At that time, there is genetic rearrangement in the genes encoding the immunoglobulins or the antibodies, and unique to that particular type of antigen which has come inside my body, a specific type of antibody will be synthesized. So these antibodies or the immunoglobulins, they will counteract with these antigens with great specificity because of the complementarity in their structure. They will counteract with the antigen with a high amount of specificity. Normally, these proteins or the glycoproteins, they have a molecular weight of which ranges from 150,000 Dalton to 900,000 Dalton. Now, these antibodies, they can function as the effector of the humoral response by binding to the antigen and neutralizing it or facilitating its elimination from our body. So, as you can understand from this sentence, that when the antibody is interacting with the antigen, there can be different mode of action, different mode of immune response. Either there can be the neutralization of the antigen itself or the elimination in an indirect process with the help of other components of the immune system. So when an antigen is coated with the antibody, when this interaction is happening, physical contact is there between the antigen and the antibody, it can be eliminated in several ways. There are three instances that we have mentioned over here. Number one, the antibody, they can cross-link more than one antigen. And when these several antigens are cross-linked, a cluster or clumping-like structure is formed, which is much more readily ingested by the phagocytic cells. This type of antibodies, they are also known as opsonin, and we will have the idea about them in our later sessions. Secondly, the binding of the antibody to the antigen on the surface of a microorganism can lead to the activation of the complement system, either by the classical pathway or by the alternative pathway. And it can lead to the formation of a very efficient component, which is known as the MAC complex or the membrane attacking complex. And that membrane attack complex can lead to the lysis of the foreign microorganism. Thirdly, the antibodies can also neutralize the toxins or the viral particles by coating them. Because of the coating of these viral particles or the toxin with the antibodies, their binding site becomes masked. And when their binding site becomes masked, these antigens fail to bind with the specific target site through which they will be internalized by the cell, a specific receptor. So as they fail to interact with the receptor, these toxins or the viral particles, they will not be internalized by the cell. They will not be able to enter inside the cell, which will prevent them from causing the harm to the cell or causing different disease manifestation. So these are the different strategies by which the antibodies influence the humoral immune response. If we look into this picture at the very bottom, this is the pictorial representation of how the B cell activation occurs leading to the formation of the antibody. So initially, the naive B cell, it will interact with the antigen for the first time with the help of the membrane-bound immunoglobulin. Okay. So once it has interacted, they can bring some alteration in the structure of the antibody, structure of the antigen by phagocytosing it, and then they can present a portion of this particular antigen to the T helper cells with the help of the class 2 MHC molecule. So here the B cells, they are acting as the antigen presenting cell. So once these activated helper T cells, they become active, they give rise to a large number of different types of cytokines, which are actually growth factors in nature. And these cytokines, they can act upon the B cells, activating them, activating the process of their clonal proliferation, leading to the formation of two different populations of cells. So the first population is the memory cell, which looks like the mother cell itself. And we have another population of a larger size cell, and they are the plasma cells. Now, these plasma cells, they have the protein secretion machinery in them. They do not express the surface-bound immunoglobulins to that extent as the mother cell. Rather, they have the protein synthesis machinery, the endoplasmic reticulum, the Golgi bodies, and the other components. With the help of that, they can encode for the particular antigen-specific antibody. They can produce the antibodies from them, and they can secrete out the free antibodies for in the circulation. So this secreted antibody in the circulation, when they come across or when they confront these antigens, they interact with them and neutralize or eliminate them from our system. So this is the basic process of activating the immune response or triggering the immune response in response to a foreign invader or a foreign particle in our body by means of the antibody. So let's have an idea about the structure of the antibody. Now, this antibody structure, let us look into this particular picture. It consists of 4 different chains. Okay, so it is made up of 4 polypeptide chains. So if we refer to this particular picture, 1, 2, 3, 4, 4 polypeptide chains are present in this structure of the immunoglobulin. Here we have 2 identical light chains. These light chains, they have a molecular weight of approximately 25,000 Dalton each, and they are approximately 220 amino acids long. And we also have 2 identical heavy chains of the 8 chain. They are larger polypeptides of almost 440 amino acids long, and they are 50,000 Daltons or more in molecular weight. So if we go to the picture, so here we have the light chain, this is another light chain, and these 2 are the heavy chains. So this light chain will interact with one of the heavy chains, and the other light chain will interact with another heavy chain, and they form this tetrameric structure. So each light chain is bound to a heavy chain by means of the disulfide bond, and by such non-covalent interactions like the hydrogen bond formation, hydrophobic interaction, or short link formation, to form a heterodimer of one heavy chain with another one light chain and another heavy chain with another light chain, the HL dimer. So one HL dimer will now interact with another HL dimer by means of the non-covalent interactions and the different types of disulfide bridges. So these non-covalent interaction and the disulfide bridges, they will link the 2 identical heavy and light chain combinations to each other to form the basic 4-chain antibody structure, which is also referred to as the dimer of dimer. Why dimer of dimer? Because here we are having the first dimer. Let's go to this particular picture. So this is the first dimer we have. In the left hand side, the heavy chain and the light chain, they are interacting with each other. This is one of the dimer. The second dimer is this second light chain, which is interacting with this heavy chain. This is in the right hand side. So one of this dimer and the other of this dimer, they are interacting with each other to form the dimer of a dimer. Okay. Now, this structure of the antibody comprising of the 4 polypeptide chain, it can be divided into two segments. Okay. The structure, as you can see in this picture, it looks like the Y alphabet, and the stalk of this Y alphabet, it is referred to as the FC region. Why? Because this FC portion of this region of the polypeptide chain, that can be crystallized. That's why it is referred to as the FC region. And the top most arms of this antibody, which is also looking as the arms of the Y alphabet, that is referred to as the fragment antigen binding, because this is the portion actually which is interacting with the antigen. So the FC region has no role in antigen binding. It is the topmost two arms which have a role in the antigen binding. So these two arms, they are referred to as the Fab arm. This is one of the Fab arm. This is another Fab arm. So altogether, these two, they form the Fab2 complex. So here we have Fab2 and here we have the FC region. Now, in both the heavy chain and the light chain, we have the first 110 or a little bit more or less number of amino acids in the amino terminal region that varies greatly among the antibodies of different specificity. So this particular region is known as the V region or the variable region. So in case of the light chain, the V region will be referred to as the VL. In case of the heavy chain, it will be referred to as the VA. Now, all of the differences in specificity that is demonstrated by different antibodies, it can be correlated with the differences in the amino acid sequence which is present in this variable region. So more and more variety we have in the amino acid sequence of the polypeptide chain, more will be the uniqueness of the region that binds with the antigen. And because of this uniqueness, we will have diversity in the antigenic specificity. Now, most of the differences that among these antibodies, the different types of antibodies which are formed in our body, the most of the differences and most of the unique character is concentrated within a particular zone or within the cluster of zones of this V region, and that is referred to as the complementarity determining region. So in the variable region, as I mentioned, there will be the variation in the amino acid sequence, and within that variation, again, there are certain regions where most of the variability is concentrated. And those regions are actually the regions which are interacting with the antigen, and they are referred to as the complementarity determining regions or the CDRs. And the regions of the relatively constant sequence, that means the amino acid sequence will be more or less the same. Okay, so the regions of the relatively constant region beyond the variable region, they are known as the C region or the constant region. So they will be conventionally referred to as the CL on the light chain and CH on the heavy chain. So in the total structure of the antibody, the variable region will be comprising of VH and the VL. So again, if I refer back to this picture, so the variable region, it will be comprising of the VH region and the VL region corresponding to the variable region of the heavy chain and the light chain. And we will be having the constant region which is further segmented into different subparts. And they are the CH1, CH2, CH3, and sometimes it is CH4. So in one of the in the light chain, we have one variable chain region and one constant region. And in the heavy chain, we have one variable region, no doubt, but based on the structure of the antibody, based on the amino acid sequence that is present in this particular junction, which has been marked as black over here. Based on the amino acid sequence over here, these constant regions, it can be either 3 in case of the immunoglobulins, or it can be 4. So it can be either CH1, CH2, CH3, or it can be either CH1, CH2, CH3, CH4. Now, if the antibody does not have any hinge region, this black region is referred to as the hinge region. If the hinge region is not present, there will be 4 segments of the constant region, C4. And if the hinge region is present, then there will be 3 constant regions, CH1, CH2, and CH3. Now, this particular portion, that is the constant region, is attributed to the biological activity, and the topmost portion of these 2 Fab arms, they are dedicated for the antigen binding. Now, how the structure is actually looking like, and which structure is having, which component of the structure is having which particular type of function. For that, different types of experimentations have been done. So initially, it was checked which portion is having actually the antigen binding capacity. Okay, so for that, since they are proteins or glycoproteins in nature, they can be digested by different proteases if those proteases have their recognition site in this particular polypeptide chain. So when these antibody, IgG antibody was first taken as the sample, and they were treated with the papain, it was found that they are breaking down into 3 segments, right? And these 3 segments, again, they can be segregated into 2 parts based on their molecular weight. There were 2 identical fragments and 1 fragment which is different in their size. So when the gel was run after breaking down the protein into its segments, then we had 2 bands identically at one position and another band at the different position. So here from we got an idea after the papain digestion that there are 2 identical components and 1 different component. And these 2 identical components are the parts of the Fab2 arm. That means there are 2 Fab arms present and 1 Fc region. Again, the same type of experimentation was done where papain was replaced by pepsin. And here, after the pepsin digestion, it was found that there was a band found which is double in size, double in molecular weight than the 2 identical arms that we got after the papain digestion. That means pepsin is digesting this antibody in such a position that these 2 arms are remaining interlinked, that is beyond the hinge region. So these 2 Fab arms, they are being linked by these disulfide cross linkages. So here we are getting a single component which has the molecular weight twice the size of the molecular weight of the identical components that we got after papain digestion. Again, in the 3rd experiment, it was done, what was done is that the antibody suspension or the antibody preparation, it was treated with beta-mercaptoethanol. Now, beta-mercaptoethanol has this reduction reaction, as a result of which it was found that here we are getting 4 different bands. And these 4 different bands, they are corresponding to the molecular weight of the light chain and the heavy chain. So there were 2 bands for the light chain and there are 2 bands for the heavy chain. So from this particular experimental setup with different types of experimental strategies, either by treatment with a chemical or either by treatment with 2 different proteases, we got the idea that the antibody is made up of 4 polypeptide chains. Number 1. 2 of them are identical and larger in size, so they are the heavy chains, and 2 polypeptide chains are lighter in size or smaller in size, and they are the light chains. Again, this particular antibody structure, it can be divided into two parts based on the pepsin digestion, where the antigen binding site is remaining intact in the CAP2 arm. Similarly, after papain digestion, we are getting the two components which are actually half in molecular weight to that of this CAP2 arm, and each of them has the antigen binding capacity, and another arm was found which actually has the biological activity and which is crystallizable, and that corresponds to the FC region. Now, different types of immunoglobulins or the antibodies are there. Based on the abundance of them in our circulation or in our body, they can be named as immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. And if we look into this pictorial representation, say for IgG, IgA, IgD, IgM, and IgE, we can find that they have a uniqueness in their structure, right? So here, in case of IgG, we are having a monomer. IgD and IgE, they are also monomer. But IgA, as you can see, it is present in the form of a dimer. And on the other hand, IgM, here we are having five components, five antibodies linked together to give a pentameric structure. So, as we can see that IgG is the most abundant type of antibody in our body, followed by IgA, IgM, IgD, and the least abundant, or sometimes it is not at all present in a particular individual, and that is the IgE antibody. Now, if we have a detailed look into the structure of the antibody that corresponds, instead of looking into the cartoon structure that we referred to in the initial slides. So based on the type of amino acid sequence, here we have displayed the structure where the different parts of a single chain, they are linked by the disulfide linkages. So, considering the variable region or variable domain of both the light chain and the heavy chain, this picture has been taken from Immunology, Book of Immunology, written by Janice Kubi. So, if we consider the variable region domain of the light chain and the heavy chain in case of the human antibodies, maximum variation is located in those portion of the sequence which corresponds to the loops that joins the beta strands. So in this particular structure, beta pleated sheets are present, and in the beta pleated sheets where the disulfide linkages linkages are there, forming this loop-like structure that corresponds to the CDR, or the complementarity determining region. Since the variation is much more higher in these CDRs, they are known as the hypervariable region. So, the variation being concentrated in this particular part of the variable domain, they are known as the hypervariable region. Hypervariable regions form the antigen binding site of the antibody. So, this is the portion of the antibody. This particular region of the antibody is actually dedicated for interacting with the antigen, and because of the hypervariable nature of the amino acid sequence over there, the antibodies, they can show their activity against diverse form of the antigens. Now, as the antigen binding site is complementary to the structure of the epitope. Epitope is the antigenic determinant which is present in the antigen or the foreign molecule. So since there is structural similarity, the structure is complementary to each other, these regions are more widely known as the complementarity determining region, or the CDS, as we have mentioned before. The 3 heavy chain and 3 light chain CDR regions are located in the VH and the VL domain. And rest of the VL portion, that is apart from these loop structures, the other portion is referred to as the framework region, because here variation is present in the sequence, but that variation is not as high as that we observe in case of the hypervariable region or the complementarity determining region. So these are actually acting as the platform to hold up the region of the hypervariability, so that there is an optimum interaction of the antibody with the epitopes which are present on the target antigen. Okay, so the wide range of specificities which are exhibited by the antibodies are due to the variation in the length and the amino acid sequence of the 6 complementarity determining region of the Fab fragment, and the framework region, as I mentioned before, it acts as the platform or a scaffold to support these 6 loops, so that the interaction is maximum between the antigen and the antibody. Now, this is a general idea about the functions of the antibodies. First of all, there can be the neutralization. That means the antigenicity or foreignness of the antigen can be neutralized or can be depleted of by interaction with the antibody. That's why it is known as the neutralization. Sometimes this neutralization is not happening, but the antigen and the antibody, because of the complementarity of their structure, they can interact with each other to form the antigen-antibody complex, which is also referred to as the immune complex. And this immune complex also has different roles in the immune system to trigger the different arms or immunities of the immune system in one way or the other. And this immune complex formation involves different types of non-covalent interaction. Thirdly, we have the opsonization. So when I was giving you the introduction to the different types of strategies by which antibody contribute to the humoral immune response, I mentioned that the antibodies, they can cross-link with multiple antigens at a time, increasing their size. A cluster-like structure is formed, which actually welcomes the phagocytic cells towards them, so that the phagocytic activity is enhanced. So here, the antibodies, they are playing a role to enhance the phagocytic activity of the phagocytic cells, like the macrophages of the dendritic cells. So this particular process of enhancing the phagocytic activity is referred to as the opsonization, and the antibodies which play part in it, they are known as the opsonin. Again, when the antigen-antibody complex has been formed on the surface of a bacterial cell, because it has the antibody has been generated against the surface antigen of the antibody. So there, that antigen-antibody complex formation can lead to the activation of the complement pathway. And here, the MAC complex can be activated. Apart from that, different complement split products are also produced, which can act as anaphylatoxins, or which can play a very important role in eradication of the bacterial infection as well as viral infection. They can also cause the disruption of the cell membrane or rupturing of the cell membrane by insertion of the MAC complex into the plasma membrane of the bacterial cell. Next, we have the antibody-dependent cell-mediated cytotoxicity. Now, in our bodies, as I mentioned, that there are different types of lymphocytes present or different types of immune cells present. One of the such cell is the natural killer cell. And this natural killer cell, they can show the toxic effect on their target cell, which is antigenically proclaimed, right? So, what happens is that the antibody will come and interact with the antigen. It can be either a viral particle or it can be any transformed cell, malignant transformed cell, which is expressing the surface antigen on their surface. So the antibody has been produced against one of the surface antigens over there, and it has come and formed a complex over there. Now, this natural killer cell, it has one particular component on its surface, that is CD16, with the help of which it can interact with the Fc region of the antibody, particularly IgG antibody. Okay, So we have already seen that this Fc region is for this kind of biological activity. Right? So it can interact with a specific receptor which is present on the natural killer cell. So now here the antibody is acting as a bridge between my target antigen and the natural killer cell. And it brings the natural killer cell in close proximity of the antigen, which maybe otherwise would not have been possible. So since it is targeting the cytotoxicity of the natural killer cell on the tumor cell or the virally infected cells, this type of process is known as the antibody-dependent cell-mediated cytotoxicity or ADCC. Sometimes there can be the hyperproliferation of the B lymphocytes. And whenever there is a particular defense mechanism present in our body, so there should be some kind of discipline over there so that each and every component of the immune system is actually functioning in a disciplined way, in a optimum way, so that only adequate amount of component is produced, only adequate amount of arms and immunities are produced by the immune system. But if there is the overactivation of the immune system, then it can give rise to the hypersensitivity. That is an abnormal manifestation of the immune system. And in this hypersensitivity also, the overproliferation of the B cell or overexpression of the antibodies, that play a very important role. Apart from that, there can be the immunodeficiency. That means there can be either some kind of genetic manipulation, there can be some kind of mutation or suppression, as a result of which the B cells may become depleted or the antibody production may become inhibited, which can give rise to different immunodeficient diseases. And sometimes antibodies may be formed against my cells molecules. So if antibodies are made against my self proteins, my own components, so that will cause destruction of my own cells, and this comes under the autoimmunity. So these last 2 points where we are having the function of the antibodies, these are the abnormal functionality if the antibodies are overexpressed or downregulated. And rest of the 5 activities that we have shown, that is the normal activity of the antibodies in diverse ways to help the immune system to combat the effect of the foreign antigen. So today we are ending over here. We will come back with the second session of the antibodies shortly. See you then again. Bye.